The Open Bioeconomy Lab has identified a list of DNA polymerases that are off patent due to patent expiry, and we're now looking to take those sequences from the patent, code and optimize them for expression in E. coli, and remove any problematic restriction enzyme binding sites, particularly those used in DNA assembly techniques that are standard practice in the synthetic biology and biotechnology field. So here's our list of DNA polymerases. We're going to start with BST DNA polymerase full length. As you can see, we have identified the original paper. We've also got links to the patents, and the US issue date of this patent was February 23rd, 1999, following a filing in 95. So due to US rules around patent expiry, we've identified the expiration date as April 12th, 2011. Using the lens, we've identified sequences within the patent so that the lens has helpfully already automatically extracted for us. And so I'm just going to click through to that now. So one thing to bear in mind with the lens's sequence extraction is that it still requires some manual checking to establish which sequence is the actual sequence of the enzyme that you want to use, as opposed to another sequence that has been included in the patent. And so here you can see the DNA sequence for what we believe is BST full length enzyme. And so you can see here, importantly, it is mentioned in the claims, which means that it's actually what the patent is covering as opposed to something that's being referenced as background material. So just if we look at which sequences lens.org has pulled out, you can see here they've identified 34 sequences by text mining. And so we can use some filtering techniques to identify what the actual sequence we want is. If you scroll down here, we're initially looking for a DNA sequence, although we can come back to that. We do not want any nucleotides that are under 100 base pairs because that indicates that they're not in fact full enzymes. So we're going to restrict by that. We're also only interested in sequences initially that are located within the claims of the patent itself and not within the background material or the detailed description. So we're going to refine by all of these factors here, and we end up with 7 sequences. Okay. So as we scroll down, you can see that they're all reasonable length, which makes it quite difficult to identify from this information exactly which one is the enzyme sequence that we want. So we're going to clear this for a second and try a different technique, which is to look for peptides or protein sequences instead. We want it to be over 300 amino acids. We want it to be located in the claims. And then if we refine by that criteria, we end up with only one sequence. So this is looking more promising. We've only actually got one sequence. It's cited in the claims. It's a full-length protein or peptide, and so we can be reasonably confident that this is the correct one. However, we should go back to the original document and just check that and read the claims themselves to make sure. So I have already done that. I'm not going to display it on the screen. This means that one of the ways in which we could identify the DNA sequence, if the pattern is written confusingly, sometimes they are, is to just translate the DNA sequence into a protein sequence and compare the two. In actual fact, if you go back to the original patent and read it in depth, it becomes clear that the DNA sequence we want is number 21, which is what we had previously. So I can go, I'm just going to clear all of these and go to sequence ID number 21. We will do a second check on this, so we will make an alignment between 21 and the protein sequence we just identified. That didn't quite work. 21, refine. Great, so this is the one that we want. If we click on the DNA symbol here, we can actually go back to the page we originally started on, which has the FASTA sequence that we can copy and paste, copy to clipboard or download using the tools that Lens provides. Okay, so we've imported this into Benchling already. So if we go across to here, you can see that we have the full-length DNA sequence. Benchling has a number of tools that will be helpful for us here. So if we go to the settings, we can choose what annotations we want to see. We can also create new sequences and translations of this DNA sequence. So in Create, we can translate forward, which is what I've already done. So you can see the coloured amino acids there underneath their corresponding codon. We can also produce new protein translations. New protein forward will give you this type of file here, which is the full-length DNA translation for the bases selected. So you can see that here. Now, one final check we're going to do before we proceed with the sequence is check the translation of our chosen DNA sequence to the amino acid sequence that we identified in the claims of the patent. So I've already copied that up here. That's a direct copy from the FASTA file at the Lens. And so we want to create an alignment of those two. So in Benchling, we come across alignment, create a new alignment, and then we want to add it in here. So I'm just going to start typing the patent number, and hopefully it should come up. Okay, so now it's come up with the other amino acid. So we're only going to align two, so we select both, create alignment using Clustal Omega. It takes a short period of time. It's a reasonably short sequence, and there's only two of them. But Benchling will notify us when it's finished. Okay, so the alignment has been successfully created, so we'll now open that. And as you can see, there are 876 identical sites. The pairwise identity is 100%, so they're exactly the same, which is positive that we selected the correct DNA sequence for the amino acid sequence claimed in the patent.